Good afternoon. My name is Gina Wen. I'm a SMID-cap biotech analyst. It's my great pleasure to introduce our next speaker, Simon Harnest, VP Strategy of Finance from Cellectis.
Thanks so much, Gina. Thank you again for having us. Beautiful Miami. Very sad to be flying back to New York tonight. But I'll make this very short so we have enough time for Q&A. It's a little longer presentation, but we can always follow up after if any one of you have more questions. We can do this one-on-one or in the breakout session. But I'm very happy here today to present Cellectis, an allogeneic CAR-T company, and one of the first in its space. And I will talk a little bit about our platform technology and also about our pipeline and what to expect in terms of data flow this year, and also maybe wrap up with where we see the space going. So just as a summary, Cellectis is really built on gene editing. So the company was founded in 1999 and was one of the first companies to really explore gene editing as a therapeutic potential in healthcare, in different programs. And we started with different technologies and notably meganucleases in the beginning. We explored all other gene editing technologies out there, including zinc finger nucleases, TALENs, and CRISPR. And today we have a very broad set of patents in the TALEN gene editing space, which we believe is one of the premier tools to really precisely modify the genome of cells. And we built a pipeline of allogeneic CAR-T programs, which we believe is on the forefront of oncology today. We've seen notable advances with autologous CAR-T cells in the oncology space by companies such as Juno Therapeutics or Kite Therapeutics, and also Novartis, who actually has the first commercial approval for CAR-T. And the phenomenal response rates here are really what drives us into the space, but from a patient access perspective and also the viability of the product can vary greatly on a patient-by-patient basis. And that's why we decided to use our proficiency in the gene editing space to basically develop a program, what we call allogeneic CAR-T or off-the-shelf CAR-T. So these are T cells that are sourced from healthy donors that are then modified genetically and can be dosed at any patient around the world. So I explained some of those differences already, but notably it's like you have to think about this. A patient comes into the clinic in an autologous approach at a very critical stage already, and they source the cells from that patient, modify them, and then filter them and put them back into the patient for treatment. And this entire process can take somewhere between, I would say, 6 weeks and a few months because the manufacturing times are very lengthy. Also, you don't know if the quality of the T cells you get from a very sick patient will be good enough to have a significant anti-tumor effect at the end. And all this while the patient is potentially progressing in the disease and waiting for the cells to come back. And we've seen the price tags attached to these treatments, which can be pretty significant. The prices are north of $400,000 just for the treatment, and not including the time the patient has to spend in an ICU, for example, or the outside treatment to contain side effects can be pretty expensive, up to $1 million. And with an off-the-shelf approach, you circumvent a lot of these difficulties. You basically produce cells up front. So you go kind of through the same manufacturing process that you would do on a patient basis in the autologous process, but you do this with a much higher cell number of starting material. You know that these cells are coming from fit and healthy donors where the T cells have their full potency. And you do this in quantities that after one manufacturing run currently, today we get a few hundred doses per manufacturing run. So we can potentially treat a few hundred patients with just one manufacturing run, which takes about 2 weeks to produce. But this manufacturing is basically on an ongoing basis and is followed with a very strict and thorough quality assurance process. So this process is something that can take a few months, but you have to think that the patient doesn't wait for the treatment for a few months, because by the time a patient gets accepted into a trial or would be ready for treatment, these cells have already been qualified in terms of their potency, quality, and the cleanness of the product. And again, this lowers the cost of goods per patient significantly by logs. And we have given publicly a number of about $4,000 per patient in terms of cost of goods currently, and we're still optimizing the manufacturing process in terms of more cells we can handle at the same time. I briefly mentioned TALENs. So this is our gene editing tool. What you see here, it's a graphic that depicts the TALEN mechanism and how it looks. So these are basically two pairs of 16 RVDs, repeat variable D residues, that are assembled to recognize a specific base pair. And the precision of this is really very outstanding. At, I believe, 6 to 7 base pairs specific, we can hone in on one target. Very impressive efficiency of this tool. Also, the rates of knockouts are routinely over 90%. In GMP environment, in R&D, we get over 99% single knockouts. What's also very interesting for future products potentially is the knock-in rate, or the efficiency at which we can insert a genome, which is basically a consequence of a clean cut and homologous recombination of the genome, and we do this at over 80% efficiency with a single knock-in. I will skip a few slides to get to our partnerships here. We have a partnership notably with Servier and Allogene. And I will explain this a little bit because it's kind of like a triangle relationship. Going back to 2014, Cellectis partnered a lead program, UCART19, with Servier for exclusive worldwide rights to Servier. And Servier then later that year in 2014 sub-licensed UCART19's US rights to Pfizer. At the same time, Cellectis did a deal with Pfizer on 15 targets. And so at that point in 2015, Pfizer had access to 15 targets potentially licensed from Cellectis, as well as the US rights to UCART19. In 2017 then, or 2017-18, Allogene, as a new company, took over the portfolio of programs from Pfizer. And we're very excited about this collaboration. Allogene is built by the former management team of Kite Therapeutics, with Arabelle de Grün and David Chang at the lead. And that program is really moving along very, very fast. UCART19 is the lead program there, and I will come back to it a little bit later. Pfizer still has an equity stake both in Cellectis and in Allogene. Coming to our pipeline, I have highlighted here in blue the programs that are Cellectis' proprietary assets. And I will focus on these in this presentation because this is really what's important for the news flow for Cellectis this year. UCART19, which you see here up top, the gray bar, is driven by Allogene and Servier in Europe, Allogene in the US, and we're very excited that Allogene recently filed an additional IND for UCART19, or they call it ALLO-501 in NHL, which is the next patient population that UCART19 will be introduced in the clinic. So focusing on Cellectis' proprietary assets, this pipeline chart is not in order of chronology. It's more grouped by disease indications. And what you see here is our concept of developing a full library of off-the-shelf CAR T cells that can attack different tumor-associated antigens in specific diseases. So you really try to own a disease by going after different antigens associated with that disease. So for example, in ALL, it's CD19 and CD22. For example, in AML, you can go after CD123 or CLL1 and a few others. Or in multiple myeloma, many of you maybe know the BCMA antigen, so you can go after BCMA, and also CS1, which is one very interesting target for us at Cellectis. So maybe starting there, we picked UCART-CS1 because of its expression profile on multiple myeloma cells. It's very well expressed. It's potentially higher expressed than BCMA. And the trick here is that CS1 is also expressed on certain T cells. So when you think about making a CAR T going after CS1, you also have to remove CS1 from that T cell before you transform it into a CAR T cell. Otherwise, they would kill each other in the manufacturing process. And through our gene editing proficiency, we enabled that step, and we basically remove CS1 from the surface of the cell before we make a CAR going after CS1. This is a program that we're very excited about to enter the clinic this year. UCART22 is another ALL program, that will also enter the clinic this year. It's a CD22-directed CAR, which is very similar to UCART19. And with this approach, again, we want to offer patients the opportunity to have another CAR T treatment, potentially following a relapse after CD19 CAR T treatments. So it's a little bit of a niche indication, but it's an interesting unmet need, because there's currently no other option for these patients that basically relapse after a CD19 CAR T treatment. And now coming to our lead program, UCART123 in AML, this is the most advanced program in the clinic. It has been in the clinic since 2017. We were briefly on a clinical hold and had to reduce the doses to start with, and we basically spent 2018 going through a slow dose escalation process that was mandated by the FDA. And now this year, we're getting back into dose levels that we think will be very significant therapeutic dose levels. So UCART123 is planned to have an interim data readout by the end of this year at a scientific conference. And we're very excited to share really the first clinical data with an allogeneic CAR T in AML, which we believe is one of the most difficult hematologic diseases to treat today when you're in the relapse refractory patient population. So I summarized a lot of details about these programs already. That's why I'm going to skip through a few slides and then quickly go to Q&A. So again, UCART19, just to summarize this, it's currently in a dose escalation study that is performed in pediatric patients and in adult patients in two different studies. But the pooled patient data so far that was presented at ASH last year showed about an 80% complete remission profile at day 28. That's very impressive, we think, for already a study that combines both the low doses and medium doses of this drug, which are still way below the currently approved autologous CAR T doses. And this program is moving forward nicely towards phase 2. And we're very excited because this was a program that Cellectis designed and built, and it's now being put into the clinic by some of the best people in the world. So maybe really quick, CD123 as a target in AML, it's a little bit of a debate what's the best target to go after in AML, because there's different targets with different levels of expression, both on AML cells and also on healthy cells. For CD123, we picked a target that is expressed on AML cells, but it's also expressed on myeloid progenitor cells, which are very vital cells for you to rebuild your blood out of your bone marrow. Therefore, we think with an allogeneic CAR T, we're hitting, so to speak, a sweet spot, because we have, on the one hand, the power of a CAR T, but on the other hand, we also have a limited persistence that doesn't span over a few months to years, but is probably limited to a few weeks, which would be enough to show an ad hoc anti-tumor effect in these patients. So we're progressing this through a dose escalation, which is the critical step for all allogeneic CAR T trials. Because when you think back how this space started with allogeneic CAR T, the biggest concern the FDA had was the risk of GVHD. And we removed this risk by gene editing out the TCR from these donor T cells. And then introducing this into the clinic, we were bound by the FDA's guidelines to really go through very slow dose escalation to prove that in patients we don't show GVHD. And we have shown this. We have currently never seen any form of GVHD in AML patients, so the drug is very safely administered. But we do see your typical profile of CRS that we're very accustomed to from the CAR T cell space, which CRS stands for cytokine release syndrome, is also a function of the activity of your CAR T cells against tumor. And patients with high tumor burden have potentially high CRS. Patients with low tumor burden generally show a lower CRS profile. And this is something that we're exploring very carefully in this dose escalation phase, where we want to see what is the optimal dose for a patient with a certain type of tumor load that will give you the best anti-tumor effect while being safely administered as well. So this is the work we're performing right now, and the goal of this phase 1 study is really to show, can UCART123 significantly reduce tumor burden in a safe manner? And then the thought process for our phase 2 would be, what is really the treatment strategy for these patients in the longer term to increase overall survival? Is this going to be a bridge to transplant, which is currently basically impossible for these patients because there's nothing that reduces their tumor burden and enables that bridge to transplant? Or can we explore in our phase 2 a multi-dosing approach to give repeat dosings of UCART123 over a few months, for example, to show that we can reduce step-by-step more and more tumor burden to a level where this could potentially be managed as a chronic disease. So these are kind of the thought processes we're pursuing with UCART123, very different in AML than it is, for example, with UCART19 and ALL. Again, I won't go too much into this, but we have a very nice preclinical profile for the product. Again, UCART22, I'll go through this very quickly. Basically, the idea is to do the same dose escalation as we've done with UCART19, relatively similar doses to UCART19, so we have a certain idea of what to expect in terms of expansion of these cells and persistence. This program being part of the ALL patient population that could potentially be pre-treated with alemtuzumab, also has the CD52 knockout. So this is something that our partner Allogene is talking about a lot, is the usage of alemtuzumab as an immunosuppressant in the preconditioning for these patients, in addition to cyclophosphamide and fludarabine. And therefore, in order to have a CAR T that's persistent in the presence of alemtuzumab, you have to knock out the CD52 receptor. And so UCART22 does have that built in. We think very critically about the usage of alemtuzumab because it really significantly reduces the immune system of these patients. But we also see that here, in this case, a longer persistence of the CAR may be needed, and therefore, with the CD52 knockout, with alemtuzumab, it's not a bad treatment approach. UCART CS1, or the target CS1, as I mentioned earlier, is a very interesting one to us because of the high expression on multiple myeloma cells, but also from a technical perspective that you have to knock it out from your T cell before introducing a CAR going after CS1. You also know this target as SLAMF7. There's actually an antibody called elotuzumab, which is an approved antibody by BMS and AbbVie, which has shown some really nice data. Actually, recently at ASH, there's been a 160-patient study performed in kind of a real-world scenario of relapse refractory multiple myeloma patients, and they've shown over 30% overall response, which for a naked antibody alone is pretty impressive as a validation for the target. Compare that to BCMA, there's not really an antibody that shows that type of response. So we figured out this technical challenge, and we manufactured this program this year. And now we're just waiting for the clearance for the clinical trials. The IND has been submitted and accepted by the FDA, and we're now looking forward to bringing this into clinical trials. And we will announce to the street when we'll have our first patient in and so forth. The protocol for this program has been written a little differently to the other programs that we started with. This starts at a little bit of a higher dose already, and the timeline from patient to patient is a little faster. So we're very excited to see this to be starting enrollment, because this could potentially enroll patients faster than the other previous programs, and could also be a guideline for the FDA for future faster enrollment schedules. So again, I explained this already, that you have to knock out CS1 from the T cell before introducing a CAR going after CS1. So just to sum it up really quick, this year we are potentially in the clinic with 3 programs. We're currently in the clinic with UCART123, and we will potentially add 22 and CS1 in the next few weeks. And then we would have a much more vital patient enrollment schedule and news flow on the clinical side than we had before. And this is really the time for Cellectis to go into the clinic and to show what the proprietary pipeline looks like. And I think, just like as a thought behind this allogeneic concept, This is really something that we built in-house at Cellectis. This doesn't come out of a clinical center, such as the NCI with the autologous programs for Kite, for example. And here we're really building a program as a company, and we're building not just one lead program with UCART19, but actually a whole pipeline of different indications and programs we're going into the clinic. So it's a very exciting time for us because I feel like the hard work has paid off on the manufacturing side. We were actually able to produce not just one, but in total 4 programs that are going to be in the clinic this year. So I'll skip some of these gene editing slides and give Gina more time to ask questions. I think we should wrap it up here. I think we don't have that much time.
Thank you very much, Simon. I think maybe I would just ask one question and then save the rest to the breakout session across the hall. So I think a UCART 1 to 3 in AML. You also comment on, possibility of a redosing, and then right now, the clinical hold already moved. Are you back on track? Can you give us an update on which stage you are, and then, what is the plan? Because our understanding, you can dose like 3 different cohorts, different doses, right? So like, what is the plan? Under what condition you will do the redosing?
Exactly. So it's a very good question to understand a little better where we are at in dosing right now, and I'll just go back really quick to the slide here. So this slide only shows the picture as of today, and it doesn't show what happened in 2018 and 17. So in September 2017, we started this trial at a dose of 6.25 times 10 to the 5th cells per kilogram. That was the starting dose, 625, 10 to the 5th. We treated 2 patients, 1 AML, 1 BPDCN patient. We had a patient death with the BPDCN patient, which was due to the very frail nature of the patient. He should have probably not been enrolled in the study. After that patient death, the FDA put us on hold to really understand, is there a specific toxicity associated with CD123 in the setting of AML? There's been a lot of bispecific trials on hold with CD123 as the target, so the FDA just wanted to know what happened exactly. It gave us a very interesting insight into this trial because we saw in both patients a very significant tumor reduction with UCART123 in a very short amount of time. And however, having this as a silver lining, the reality was that after the hold, we had to start at very low dose levels. So we basically spent the entirety of 2018 until the 3rd quarter of 2018 to go through low dose cohorts at 10 to the 5th cells per kilogram, at 10 to the 4th cells per kilogram, about 50 to 60,000 cells per kilogram. That was a very low dose, and it showed that the product is safely administered in these patients, but we also wanted to go back to therapeutic dose levels, of course. And we had to follow this with a timeline of 6 weeks between each patient at a very low dose level, so it took a while to get through this. Now we are at 4 weeks between each patient, and we are back at a dose of 2.5 times 10 to the 5th cells per kilogram. So 2.5 10E5 is the current dose. We have already dosed patients at that dose. We are probably doing 2 or 3 more patients at that dose before going back to 6.25 10 to the 5th. And again, the plan is again to do between 2 and 5 patients at that dose cohort, and there is the potential to go to 5.05 10 to the 6th cells per kilogram. So there is a step up of dosing. That's one part. The other question is redosing. And we've had this question already in our UCART19 trials, where there were a few patients, I think 3 or 4 patients that have been redosed with UCART19 in ALL after a relapse following the initial treatment. And all of these patients basically got back into complete remission. And so this redosing was not part of the clinical protocol, but was on a compassionate use basis, that the doctors basically requested another dose, and we gave them another dose. Here, the consideration is the same, that you want to go through this dose escalation and find your therapeutic dose first before you really go into multiple dosing, because from a statistical perspective, from your readout of this trial, you want to first focus on what is the right dose, and then what can a redosing strategy do as a potential benefit. So this may be something reserved for our phase 1b or phase 2 study, but it's really something we're thinking about, because redosing, we think, is the real potential of allogeneic CAR T.
Mm-hmm. So for the patient, like say you have now at the dose escalation phase, right? For the patient, say in case the patient relapse, are they allowed to receive redose under current protocol?
They are. That's effectively right. The redosing is allowed from a compassionate protocol perspective, but it's not built like strategically into the protocol.
Okay.
So the doctors could request another dose, but it's not something that we have set redosing scheduled. So
I see. Okay. Thank you very much, and then we will go to across the hall, do the breakout session.
Thanks, Gina.
Thank you.